This podcast is part of a special series highlighting work to be discussed at the 2024 Cancer Neuroscience Symposium, which is hosted by MD Anderson Cancer Center and Wiley on February 28th through March 1st, 2024, with options to attend virtually or in person. Learn more and register at mdanderson.org/Cancer Neurosymposium.
Hello everyone, and welcome back to Beyond Cancer. Today, as part of our 2024 Cancer Neuroscience Symposium series, I'm with Dr. Marana Mint, and we are interviewing Dr. Humsa Venkatesh.
Dr. Venkatesh is an assistant professor of neurology at Brigham and Women's Hospital and Harvard Medical School. She has a groundbreaking work on the interactions with the nervous system and cancer, particularly looking at the central nervous system, both for primary brain cancer and also brain mets. It's a great privilege to have you here today. We're really excited to also have you in the meeting. And thanks for joining us today, Humsa.
Of course, I'm excited to be here. Thank you for the invitation.
Yeah, of course. And to start off, just to let our listeners hear about it, can you tell us a little bit about yourself and your research?
Yeah, of course. So as Moran just mentioned, I am an assistant professor at Brigham and Women's Hospital and Harvard Medical School. My lab is almost 2 years old now, and largely what we study is understanding, as the reason why we're all here, I guess, is really understanding the role of the nervous system in the context of malignant tumor growth. And so a lot of my past work has been focused on primary brain cancers, predominantly high-grade gliomas. But as I start my lab and go into a lot of my more independent work, we're really thinking about how these non-brain derived cancers, or these non-glial cancers, both in the context of brain metastases, but also in the context of other peripheral tumors that occur outside of the central nervous system, can similarly integrate these neural inputs. And so we largely study kind of these reciprocal interactions between the nervous system and these various different types of cancers, both in and outside of the brain, with the ultimate hope, of course, of finding therapeutic targets that can ultimately be translated into the clinic.
That's great. I'm familiar with your work, and as a really top-notch scientist in this field, can you tell us a little bit about your perspective on cancer neuroscience as a field? Why do we see, its emergence recently, and actually what drove you?
Yeah, absolutely. That's a great question. I think it's a really exciting new space, and largely the reason why I think it's blossoming, and a lot of the reason why it drove me particularly into this space, is really that the time had come. I think we just happened to be in a moment where neuroscience in and of itself has really expanded to the point where there's new technologies, new innovation, new scientists that are interested in this interdisciplinary research that we can harness and utilize all now in the context of cancer. And in the past, I think, cancer research, as we all know, kind of happens in eras, essentially. And when I started a lot of my training, the focus is really on sequencing technology. It's really understanding the molecular landscape, genetic, epigenetic abnormalities that contribute to malignant growth. And then it kind of turned a little bit more into the tumor microenvironment, largely in the context of, the immune system, harnessing the immune system and the power of immune cells to either promote or inhibit, tumor growth. And then in the context of the nervous system, it just seems, especially in primary brain cancers, seemingly obvious yet largely under-recognized in the context of the role the nervous system can play in tumor growth. And so that's kind of what brought me to understand and want to investigate this really interesting topic. And as we get into it, I think just initial studies from a lot of us working in the field have illustrated really what a dominant role these nerves can play through multiple different mechanisms in the context of the tumor microenvironment. And so it's just a really exciting time to utilize all of these novel technologies in neuroscience, apply them to cancer to really appreciate this completely novel component of disease pathophysiology.
Going off of that, I did my own research, and given that your lab is focused on brain metastasis and studying the microenvironment within the brain, can you briefly explain some of the mechanisms by which electrical inputs facilitate metastatic colonization?
Sure, absolutely. So a lot of what we have done is really indication for the first time that these brain metastases can actually integrate neuronal input. And so that we're working on really understanding the detailed mechanisms through which this occurs. We actually had to take a step back, really from step zero, essentially square one, thinking about just at baseline, does activity in and of itself drive metastatic growth in the same way that it does for these primary brain cancers? And so what we now know really is this idea that neuronal activity can drive proliferation and invasion of these metastatic tumor cells. They can form, direct interactions between healthy neurons. They can depolarize in response to neuronal activity, both glutamatergic as well as GABAergic inputs. But the detailed mechanisms occurs in the context of, various paracrine signaling factors, the voltage-sensitive downstream signaling mechanisms that drive growth, the direct synaptic interactions. A lot of those questions are actually still unanswered. We're really just at the tip of the iceberg answering some of these ideas as to whether these non-glial-derived tumors can, similarly rely upon this neural input. And the answer appears to be yes in most contexts, dependent upon the tumor type, of course, but the specific mechanisms are surely going to be very different dependent upon the tumor type and the tissue type of origin. And so there's lots to understand in that context still. So I wish I had a better answer as to the exact mechanisms that were at play here, but I think we're still in the context of understanding that. But what is clear is that neural activity itself is really similarly important in the context of the tumor microenvironment of these other types of brain cancers as well.
Yeah, thank you for that. And then, if we shift toward talking about neuron-glioma communication, can you explain how bidirectional neuron-glioma communication works and how your lab studies the spatiotemporal dynamics of these circuits and throughout the tumor development as well?
Sure, yeah. So again, as I mentioned kind of earlier on, I think the reason why cancer neuroscience is emerging as such an important field is this idea that it's playing a more prominent role than I think any one of us could have really appreciated to begin with. And it's, as you mentioned, this very reciprocal interaction whereby we know that the nervous system not only drives and facilitates tumor growth, but in the other bidirectional context, these tumors can essentially, induce hyperexcitability of the neighboring neurons, thereby potentiating all of these various mechanisms of activity-mediated growth. And so we haven't focused on too much of those reciprocal mechanisms in the context of gliomas, my lab in particular. But the field certainly has made a lot of strides in understanding those mechanisms. And so we know that gliomas can actually remodel that neuronal microenvironment in multiple ways. The first one that was discovered quite a while ago, a few decades ago, and we've thought about it a lot in the context of both tumor growth as well as neurotoxicity, is the secretion of glutamate itself. And so tumor cells, or glioma cells at least, are capable of secreting glutamate and therefore inducing hyperexcitability of these neurons in the tumor microenvironment. Work from, and that was largely led by Harold Sontheimer's work. Really recently, Ben Deneen's lab, as well as Sean Harvey-Jumper's lab, have illustrated that there are specific populations of glioma cells that can actually secrete synaptogenic factors. And so glioma cells can actually secrete proteins that induce synaptogenesis between neurons in their microenvironment. Again, another mechanism that facilitates this neuronal hyperexcitability. And the last mechanism, again, that was largely led by Sontheimer's work, was this idea that intraneurons actually decrease in their number in the glioma tumor microenvironment. And so there actually is a reduction of the number of inhibitory neurons in the tumor microenvironment, which in and of itself, again, allows these excitatory neurons to fire more often. And so there's multiple different angles through which these tumors can very clearly hijack mechanisms of normal neuroplasticity to strengthen synapses and induce higher numbers of action potentials that facilitates the various mechanisms through which these neurons can actually drive glioma growth. So it's a really intriguing kind of axis of communication, and there's good news, I guess, lots of different areas that we might target in order to kind of harness the power of these neuron-cancer interactions.
Yeah, so, just continuing on your last point, and you also mentioned the piling evidence that come from Sean's Harvey Jumper and your work was a postdoc and now is a PI. And there's a lot of evidence on why the nervous system is important and how it affects the tumor growth and progression. What are the major roadblocks for moving it to the clinic at that point?
That's a great question. I think there are a number of different roadblocks, shall I say. I think the first of which, I think most obviously, that the nervous system is so powerful in driving tumor growth, but I think the reason why that's the case is that it is the master regulator of almost, every process in the human body, and the function of every organ, and the function of down to the cellular level, the function of every cell. And so broadly targeting the nervous system in and of itself is largely going to have a lot of side effects that may be, quite deleterious in the context of the clinic. And so what we really need to focus our efforts on, I think, are really understanding the specific neuron to malignant cell interactions that might be more therapeutically actionable, that would give us a leg up on specifically targeting the interactions that are most important, at least for these malignant cells. I think that can come in the form of looking at potential receptors that are amplified in the context of cancers, or different ligand receptor interactions that are most important for cancers, paracrine signaling factors, direct ion channel communication. There's lots of ways in which we might focus on, patterns of neural activity that are specific to the malignant context, different things that I think are more obvious to treat in the context of the diseased brain versus the healthy brain. Getting there, I think, may take a little bit of understanding in the context of both really understanding these specific interactions, but also teasing out, how important different neural subpopulations are for different tumor types. That's another big question I think we have yet to address. I think we're all eager, and really do want to translate this into the clinic as soon as possible. And there are ways that we can do that, but we really have to think quite carefully about the different mechanisms that we're targeting, because from what we know, these various different neurotransmitters and different ion channels and different receptors can actually act very differentially based upon the tumor in which we're really trying to target these interactions. So that's going to be one big hurdle. And the other big hurdle is, I think, really, in the context of clinical trials, really understanding exactly what our biomarkers and endpoints are. As we kind of use these neuromodulatory agents to, modify these interactions, what exactly will we be looking for in the clinic? I think really setting those benchmarks is going to be important, to think about how we might look at the efficacy of these clinical trials. And so there's a number of different things we need to, start thinking about. And together as a team, I will say the field has been very collaborative in having, a lot of different meetings, such as these, that we're going to be having in Texas very soon, bringing together people like-minded in the field of cancer neuroscience from both the clinical angle as well as the basic angle to think about how we might most effectively translate this into the clinic. And so I think there has been a lot of movement, and there will be with continued collaboration and communication with all of us that are really coming at it from multiple different angles.
Yeah, this is really cool, and I really concur and like to echo your comment about diversity. The nervous system is ubiquitous, and you have to be very cautious when you're targeting such a fundamental system, in any host. You've mentioned the meeting in Texas. Can you tell us a little bit about the latest and greatest news from your lab and what we're going to hear about at the Cancer Neuroscience 2024?
Sure, absolutely. Well, I hope to have even more by the time that comes. But, as I mentioned, we talked a little bit about our primary brain cancers as a lot of what I focus on my past work. My lab has now moved into brain metastases, which has been very exciting, specifically focusing on small cell lung cancers and looking at how these very neuroendocrine-like tumors can similarly kind of hijack these mechanisms of neuronal input. But not only in the context of brain metastases, what we're now doing is moving into the context of the primary lung and thinking about how the peripheral nervous system, and the vagus nerve in particular, plays a central role in disease pathophysiology in small cell lung cancer in the primary lung organ. And so we're doing a lot of exciting work that I hope to, be able to give you guys updates on in the context of really understanding how vagal activity informs, malignant growth in the lung, in the primary lung, how innervation as a whole plays a role in the metastatic cascade, and thinking about, again, how innervation and the neuroimmune axis really is involved in remodeling the microenvironment to allow for this malignant growth. So there's a lot of different types of questions that we're asking that we have the capability of doing in the context of the lung, largely because the peripheral nervous system can be manipulated in slightly different ways than in the central nervous system. So I think, it's been a really fun and interesting experience to work in the brain as well as outside of the brain in the context of the peripheral nervous system, because, the tools, the techniques are very different, and the types of questions you can ask are very different. But I think you can learn a lot and apply a lot of information from one to the other. So it'll be really exciting to kind of think about how, what we find in the context of the periphery. And it's another reason why I'm so excited about, the meeting is largely because, just looking at the agenda and looking at all these wonderful speakers that are coming, I think, we're going to hear from a lot of, younger new scientists as well that are doing some really innovative work in this space. Because it's such an emerging new field, I think a lot of the investigators are, not only new and junior, but are also being able to address some of their most high risk and fun and exciting questions. And we'll get to hear about a lot of the results of that. So it'll be really fun to hear about.
Well, I can't wait to hear about all that. It sounds super exciting. It's really exciting for me to see you migrating from the CNS outside the CNS.
I probably need your help with some of that.
Hopefully. I always aspire to work with you.
Oh, please. Yes, definitely make it happen.
And I know we're excited, you're excited, and we hope you all are excited before we log off, and we'll see you guys next session next time. Bye, guys.
This podcast is part of a special series highlighting work to be discussed at the 2024 Cancer Neuroscience Symposium, which is hosted by MD Anderson Cancer Center and Wiley on February 28th through March 1st, 2024, with options to attend virtually or in person. Learn more and register at mdanderson.org/Cancer Neurosymposium.